All right, it's 3 o'clock. At least, I'm sorry, it's 3 o'clock on the East Coast of the US. But welcome to session 315, Statistical Methods for the Validation of Surrogate Markers in Clinical Trials. We have a great lineup of speakers today. I will let Nguyen start sharing her screen and pull her slides up. An is a PhD student in statistics at the University of Wisconsin at Madison.
Can you see my slides? Is okay? Just checking in.
Yes, there is this. Perfect. Yeah, looks
Okay, so
good.
all right, great. Okay, so today I'm going to talk about a project I did during kind of my master time. Um, I was advised by Dr. Sarah Lotspeich. And also, we were working with Dr. Layla Perez, and we were working on method to help correct for missing data when you're trying to do a validation of surrogate marker in clinical trial. So the gist of the talk is just we're just gonna talk through all of these buzzwords that we have in the title, and then we're gonna show how well our method is working. Okay. So in a lot of clinical trial, usually you want to see what's like the treatment effect is on some kind of primary outcome. It could be that you want to see if the treatment is working to help reduce cancer. So are you cancer free? But this can take a really long time. You might have to require patient to come to a lot of session, which is not always viable. It could really be expensive or invasive. So an alternative to this could be for us to look at the surrogate marker. So instead of trying to see what the primary outcome is so far in the future, we can just look at the surrogate marker, which sometime hopefully it's like easier to see and it's not expensive and should give you enough information about the primary outcome that's going to happen somewhere down the line. So for example, if you're thinking of cancer, you can think of tumor growth instead of actually looking at whether the patient is cancer free or not. Okay, so one framework for us that we can use to kind of validate our surrogate marker, so making sure that our surrogate marker is actually capturing all the information that we want about our primary outcome, is to use the proportion of treatment effect explained. And there are other framework that you could also use instead of this, but this is what we're going to focus on. And this is proposed by Wang and Taylor in their 2002 paper. So this is like a very specific way to do this. So imagine that we have some kind of linear regression for our outcome model here. Our Y is our outcome and our Z is our treatment group. And here we only have two, so the control versus the treatment. And a lot of the time we are interested in this coefficient beta 1 star because that's capturing the treatment effect. However, as we said before, maybe so it's now we're going to bring in our surrogate marker just to make sure that's actually capturing the information about the outcome that we want. So we can look at another regression model. And here we have kind of the same thing, but the new addition that we have is our surrogate covariates. And here we are looking at a interaction term. So we have a lot of flexibility rather than just an additive term. And what we are interested in from this model is our two coefficients that have to do with the treatment group, beta 1 and beta 2. So really the idea is that if you add your surrogate marker in and say your surrogate marker is doing a good job, so it should be capturing all the information about the treatment effect, then our treatment effect that coming from the control group should be less because we're already getting that information from somewhere else. And to kind of quantify the ideas, we would be using the proportion of treatment effect explained. So this is going to be our little r sub s. And here we define our r sub s. to be a ratio. So on the top, we have the difference in treatment effects between the two models, one where you have the surrogate and one where you don't. And at the bottom is just the treatment effect without the surrogate. So you can kind of think of this proportion of treatment effects kind of like an R square. It's kind of like that. You're thinking about how much the variation is being explained compared to without a surrogate. And this kind of makes sense. If our surrogate is really doing a good job, then that difference um, shouldn't really be that much. But if our surrogate is doing like a really poor job, then that difference will be a lot. And another thing is that since we are computing all of these number using the coefficient from the linear model that we specified earlier, this number really only work and kind of only makes sense when our model is correctly specified. So you're assuming some kind of parametric model and you're assuming this relationship, which is not always the case. And if you don't want to do that, we do have some method where we're using non-parametric models, but that's not in the scope of this talk. Okay, so our goal here is to just figure out how are we going to estimate the statistic R sub S using the data that we have. And that statistic will help tell us whether our surrogate is actually capturing all the information that we want about our treatment effect. So that's in a perfect world, we wouldn't have any problem. However, in the real world, um, we would have missing data. This is really common. It could be that your patient didn't show up for a follow-up. They could drop out due to a variety of reasons. So it's very, very plausible that we're going to have missing data when we're trying to validate our surrogate marker. So here we are going to introduce a method to deal with that in the context of trying to estimate this proportion of treatment effect explained. And for us here today, we're going to specifically assume that we have missingness in the surrogate marker value. So we have a full data set for the outcome in the treatment group, and it's only the surrogate marker that we have some missingness in. So let's just talk through how our missing data can kind of come up in situations like these. The first thing is you can have missingness completely at random, so it's independent of any covariates. This is kind of our best case scenario, and in this case, you can do a complete case analysis. This is totally fine. This is what a lot of literature do, but it's kind of inefficient because you're not using all of your data that you have. You're dropping all the observation that have missingness. The other case, which is not too bad, and we can kind of fix it, is missing at random. This is when your missingness is depending on some covariate that we know about. Here, complete case analysis is not fine. It's going to give you some kind of biased result most of the time. And the last case, which is kind of the worst case scenario, is when you're missing not at random. And for that, our method doesn't really work for that scenario. So that's something to keep in mind. And hopefully with the method that we're gonna propose today, we should be able to, when you have missingness completely at random, use the full extent of your data and gain more efficiency in your estimation. When you have missingness at random and you really shouldn't do complete case analysis, our method will give you unbiased estimator. Okay, so we are going to focus on the first and the second scenario for today. And the last one, well, it will be for our future work. Okay. And the method that I've been kind of talking about is the C maximum likelihood estimation. And this method is semi-parametric that will help maximize the likelihood of our model when our data have missing it at random. So here, you can see that we have our likelihood function for the model that we kind of talked about before. So we have the joint probability of our outcome, our surrogate, and our treatment group. And for us, we're interested in splitting it into three different models. And this, okay. So the first model is our outcome model that we talked about earlier, the one with the interaction term. This is our outcome model, and this is a parametric model that's a linear regression of y of the outcome given our surrogate and our treatment group. And this model is going to be the one that give us all the betas that we are interested in the estimation of our um, proportion of treatment effect explained. And the next piece of the puzzle is going to be our surrogate model. Maybe I went too fast, but to kind of estimate our proportion of treatment effect, we also need alpha, which are just the average expected value of the surrogate within each treatment group. And that coefficient are all coming from this model. And what's special about method is that we're doing this non-parametrically with a SIEF method. So the two coefficient that we care about, alpha and beta, that is like the the main contributor to our proportion of treatment effects. Explain is going to be coming from these two models. And the last one is going to be our treatment assignment. And if we are doing a randomized control study, we already kind of know what this is. Okay, any questions so far?
Nope, but I'll let you know you have 3 minutes.
All right, cool. So we kind of look at our simulation. If you are interested, we have a missed surrogate data where you can run these functions for yourself, or if you want to do it for your study, and you can find a result for these on GitHub below. Okay, so we simulate our data for the outcome, the surrogate value, and the treatment effect. And then once we have our data, we also simulate missingness under missingness completely at random and missing it at random. So our goal with the simulation is to estimate the proportion of treatment effect using all of the coefficient we talk about, using the gold standard. So this is where you don't have any missingness, best case scenario. Next one is complete case analysis. This is what is most often used in the literature. And the last one is what we propose, which is the SIEF maximum likelihood estimation. And to compare between these methods and how well they're doing, we're specifically concerned about the bias and the variance of this R sub S here, or the proportion of treatment effect explained. Okay. So when we have missingness completely at random, all of our three methods that we were interested in are unbiased. So this is kind of what we expected with missingness completely at random. However, if you compare between complete case analysis and SMLE, you can kind of see that we are gaining a little bit of efficiency in terms of the variance. When we are looking at missingness at random, here we're saying that our missingness depend on the outcome value. We can see that our complete case analysis is giving us biased result, which is the green box plot here. However, if you were using the SMLE method, the estimate we got would be unbiased. And finally, if we were to generate our missing at random, now not depending on the outcome, but on the treatment group, we can see that all our methods are unbiased, and especially again with SMLE getting a little bit more efficient than the complete case analysis. Okay, so just a recap since we're almost done. So we have a SIEF maximum likelihood method that will help you dealing with missing data when you're trying to validate surrogate marker. For our simulation, we can see that our method is unbiased when missing at random and gain more efficiency compared to complete case analysis. In this research, Dr. Perez and Dr. Lotspeich also look at non-parametric model and inverse proportional weight. So there's a lot more going on here that not on this presentation today. And for us, we like to, in the future, apply this method to real clinical data, like a case study, and work on how to deal with missingness not at random. And what if we have missingness in the outcome or missingness in both the outcome and the surrogate marker? Yep. Thank you to all the people that helped me with this project.
Great. Thank you so much, An. So next up, we have Emily from the University of Texas at Austin. Emily is a PhD student in statistics and data science.
Wait, are we not doing questions for on?
We don't have time to do
Oh,
questions for
okay.
An, but you can drop them in the chat.
Okay. Okay, well then I'll share my screen then. Can everyone see this?
Yes.
Okay. So should I just get started now?
Yeah, go ahead.
Okay. Okay, hi everyone. The title of my talk today is Resilience Measures for the Surrogate Paradox. And this is joint work with Lu Tian at Stanford and Layla, who's my advisor actually, who it seems like An also worked with. So a little outline is I'm going to introduce surrogate markers and what specifically the surrogate paradox is, and then get into our proposed framework and then do a real life illustration using an HIV dataset. So I think An already introduced a little bit, but a surrogate marker is usually some kind of biomarker used in a clinical trial to estimate treatment effects for costly clinical outcomes. So here as a quick example, if you're trying to estimate the effect of some kind of treatment, on some sort of pediatric care, but let's say the patients need to get a biopsy, you might want to ask, like, is there some kind of alternative alternative marker that I could do instead to circumvent taking biopsies on children? So the upshot of surrogate markers is that clinical trials are long and expensive, and if we can replace the primary outcome with a surrogate in our analysis, we can shorten trials, lower costs, or reduce patient burden. So there's lots of cases of surrogates being very good and very useful to and allowing patients to get medications that they need earlier, but it's also possible for surrogates to go wrong. So there's a very famous case here in which scientists develop a drug to prevent cardiovascular death based on the surrogate of ventricular arrhythmia. So what happened is that the FDA approved a drug that lowered ventricular arrhythmia, but it was later found out to increase patient mortality. But they only found this out after a lot of people actually died from taking this medication. And so almost all of my research centers around how can we prevent this specific case from happening. Um, so to introduce a little bit of notation, let's say we have a randomized trial. G is a binary treatment indicator where 1 is treatment and 0 is control, and let S be a continuous valued surrogate and Y be our continuous valued primary endpoint. So typically when you are evaluating the effect of a treatment on some kind of outcome, you want to find the treatment effect of G on Y. So that's the expected difference of Y between the two groups. I use delta to notate this here, and it's the expectation of Y1 minus Y0. So this is the average treatment effect. Suppose the surrogate has been validated, and I'm not going to get into the methods of validation, but there are many, and if you're curious, you can check out this paper, which introduces a lot of them. The problem is, if we see a positive effect on the surrogate in a new study, how can we be sure that the effect on Y will also be positive as well? So typically you're going to need some way to predict delta using S. So a little bit more notation is, let's define the relationship between S and Y here using these mu functions. So mu0 of S is just the expectation of Y given a particular value of S in the control group, and mu1 of S is the expectation of Y given some value of S in the treatment group. So graphically, this is what it looks like. On the x-axis, I have a surrogate, and then on the y-axis, I have the primary outcome. And these functions, mu0 and mu1, are basically just the relationship between these two variables. So our problem statement here is that current approaches generally require some form of transportability of these two mu-g functions between studies. However, this is a really strong assumption, and there's many reasons why this might not hold. So the first and most obvious one is if you have different treatments may result in different relationship between the surrogate and the primary outcome, right? You could also run into this problem if you have different applications of the same treatment, like between hospitals or even just like between different practitioners or nurses. You could also have different populations. So maybe there are some characteristics very present in this country, and then in a different country, they have very different population characteristics, and that may cause the S and Y relationship to differ as well. So our goal is to propose a framework to assess the probability of the surrogate paradox under controlled deviations of these mu functions that might be observed in future studies. Now, obviously, as a disclaimer, this needs to be combined with clinical expertise, and later you'll see why that's the case. So this is our proposed framework. So now some more notation in our specific case. Let's say we have a previous study A, and then we have a new study B. Study A has data on both S and Y, and this is where you were able to validate the surrogate. However, in study B, we have only data on S, and we do not have data on Y. So our delta value, our treatment effect in study A is estimable, but our treatment effect on study B is not estimable. So can we get some kind of probability P, which is the probability that delta B is going to be less than 0? In essence, what is the probability that we're going to get the surrogate paradox? So we can estimate the mu functions in study A, but we can never directly estimate the mu functions in study B because we don't have the Y values. So the main idea is, what if we consider functions that are similar to but not too far away from the functions that we saw in study A? What kind of delta B's would we see if those functions actually did arise in study B? So I'm going to give a bit of a visual motivation. So let's say this is the data that we observed of S and Y in the treatment group and in the control group. So this like points are the scatter plot of the observed data. So if we estimate the mu functions using whatever method, I think here we have a kernel smoother. This is the estimated value of the function in study B. So now I ask, well, what if in study B we saw this function instead? Or what if we saw this function instead? What would happen if these were the actual functions that arose in study B? Using those like new functions that might have arisen in study B, we can then construct a synthetic distribution of possible delta V values and sort of look at the distribution. So in this case, because our synthetic delta B's fall very few of them below 0, then we might say, okay, well, there's not a very high likelihood of the paradox. But in this case, if a lot of them fall below 0, then we might say there might be about a 50% chance of the paradox, so that's not super robust. So now I'll formally introduce our quantity of interest, which we call the resilience probability. So let's say for now that we have a new method to generate new mean functions in study B, which I'll call mu0 star and mu1 star. And I'll address how we actually do that later. But we can then calculate a potential value of delta B star as the expectation of mu1 star, and you evaluate this on the distribution of surrogate in study B, minus the expectation of mu0 star on the distribution of the surrogate in the control group in study B. So the randomness here comes from generating mu g star and the values of the surrogate in study B. And our resilience probability, which I'm going to call P0, is going to be the probability that this delta B star falls below 0. So visually, how does this look? Let's say you have these distribution of delta B. You're basically just looking at the proportion of them that falls below 0. So here we have it being very small, and then here we have our resilience probability is about 0.5. So how do we actually estimate this quantity? Well, instead of the true distributions of S in study B, we have just the observed values, right? So for a given value of these two new mean functions, we can calculate this is basically like the expected value of Y in study B in the treatment group, and then this is the expected value of Y in the control group, because we're basically applying the mu function to the values of S that we see and then taking the average. So again, here we're applying this potential new function to the values of S in the control group and then taking the average of that. So this is sort of our synthetic delta B star value. And then we can calculate the proportion just by taking the proportion of them that fall below 0. I also want to introduce another quantity that we care about, which we call the resilience bound. So we can consider the alpha quantile of delta B star such that the probability of delta B star lying above that specific value is 1 minus alpha. So in other words, it means that for some given alpha, you may be reasonably confident that the average treatment effect on Y is going to be greater than that value. And you just do this by taking the quantiles of the synthetic delta B samples. So graphically, if we take the example of alpha equals 0.1, what you do is you look at your distribution of synthetic delta B samples, you look at the lowest 10% of it, and then you see what this value is. So here, because our resilience bound is 0.762, we're 90% sure that the average treatment effect will be above this value, which is good because this is positive. Here, on the other hand, I look at the lowest 10% of the synthetic delta B values, and I see that it falls at negative 1.296. So we're like, in essence, 90% sure that our true delta B is going to be above negative 1.296, which is not good because that value is negative. So our proposed procedure, like written out explicitly, is as follows. So we estimate mu0 and mu1 in study A, which we denote by mu0 hat and mu1 hat. You can use whatever method you want, but in our implementation, we used a kernel smoother. So now, j different times, you generate some new mu0 star and mu1 star within a general class of functions, which we characterize as deviations from our estimated mu functions from study A. Then you calculate your new delta B star by taking the average of those functions applied on the actual S values that you saw in study B. Then you estimate the resilience probability as the proportion that fall below 0 and your resilience bound as the alpha quantile of your delta B stars. So now I want to address the question of how we actually generate these new functions. So what we want is that they use our estimated mu functions in study A as a center and stochastically deviate from mu hat from study A in a way that's governed by a set of parameters. So in our manuscript, we provide 3 classes of functions, but in the interest of time, I'm only going to talk about this first one, the Gaussian process. So in our first class, we assume that our mu0 star and mu1 star basically come from a Gaussian process with some underlying mean function and some covariance kernel. So eta0 and eta1 are some smooth mean functions, and k0 and k1 are some Gaussian process kernels. So we set eta0 to mu hat 0 and eta1 to mu hat 1 using a kernel smoother because that represents the best information that we have about what these mu functions might actually be. Then we assume a radial basis function parameterized by sigma squared and theta, although certainly you could use any other Gaussian process kernel function that you want. So we have these parameters sigma squared 0, sigma squared 1, theta 0, and theta 1 that govern the deviations in this model. So the problem here is that in each of our 3 function classes, there are unknown parameters governing the function deviation. So in that example, it was sigma squared and theta. So the solution that we have right now is that they could be fixed with clinical expertise. So for example, if practitioners believe that the 2 treatments are very different, then you could fix sigma squared g to be very high to cause the resulting functions to be very different. So admittedly, this is the biggest problem. The drawback of this framework is that you actually cannot estimate these functions if you only have one study. And so you need some kind of domain expert to tell you how to do this.
Letting you know you have 2 minutes.
Okay. So our real data application is we use 2 HIV and AIDS clinical trial data sets where our primary outcome is the change in HIV-1 RNA counts from baseline to 24 weeks, and our S is the change in CD4 cell counts. So the studies compared a 2-drug treatment arm, which is the control, to a 3-drug treatment arm, which is the treatment group. And this is the data that we see in study A. So we can see that our actual treatment effect in study A is 1.595, which is positive, and our average treatment effect on the surrogate is 70.7, which is also positive. In study B, we don't have the Y values, but we can see that the average effect of the treatment on the surrogate is positive. It's 9.5. So we use our method and we set sigma squared equal to 1.25 and theta equals to 2. These are some potential other functions. And we find a resilience probability p hat of 0.01, which suggests a very low probability of the surrogate paradox. So in conclusion, we've proposed a procedure to assess the probability of the surrogate paradox in a future study. We just assume that the conditional mean functions are randomly generated from a class of functions and generate synthetic average treatment effects in the new study to calculate the chance that it falls below 0. I have an R package which implements all of this called Surrogate Paradox Test, and the tutorial is available on my GitHub. We also have an archive link with this manuscript. In terms of future work, as I mentioned before, the biggest drawback is that the parameters for each function class needs to be set by domain experts, and only in a meta-analytic setting can they actually be estimated. So I do have a version that I'm working on for a meta-analytic setting that estimates pretty well the true underlying mean function, but we don't have a manuscript of that out yet. And something else that I want to include in the future is including covariates that might make the inference a little bit more informative based on population characteristics. So that's all. Thank you for listening.
Thank you so much, Emily. Really interesting. And so our last speaker will be Emily Roberts, who is an assistant professor of biostatistics at the University of Iowa.
Thanks, Sarah. Thanks for including me and for organizing this session. Let me know if you have any problems seeing my slides.
Looks great.
All right, thanks. So yeah, I'm happy to really kind of build upon these other two talks that were really great. It was nice to hear about some work that's going on in surrogate measures, and I'm actually going to be talking about including patient characteristics within the context of a validation method for surrogate markers. So as we've said, we're talking about surrogate markers, which we can consider in the context of randomized trials that I'll be talking about. And the idea is that randomized trials are really great, a gold standard for measuring treatment effects. Of course, they can take a very long time, so it would be great if we could use some of these surrogate measures. And these might be biomarkers, they might be intermediate outcomes. I'll be talking about time to event outcomes that tend to happen before the true outcome of interest. And of course, the idea is that the surrogate markers are somehow related to the true outcome in the trial. So again, the idea is that we'd like to use this surrogate as a stand-in for whatever a true clinical outcome is in a future trial. And the hope is that the trial could be done more quickly or more cost effectively in the future. So the idea here is that in order to use the surrogate as the outcome in a future trials, we need to be sure that it's really proper, that we can validate it and say, I feel confident using the surrogate as the outcome in a future trial. So the process here, when I talk about validation, really meaning we want to use data from a completed trial. This completed trial has data both about the surrogate and the true outcome. And we want to use some criteria to say whether or not the surrogate is a good proxy for the primary outcome. And this method I'll be talking about here is based in causal inference. So I'll say it's a good causal surrogate for the primary outcome. In particular, we'll be using the potential outcomes framework within causal inference. So I've just given a few outlines here. The notation I'll be using is the parentheses to denote the potential outcomes or the counterfactual outcomes. So in the case where we have our surrogate S and our true outcome T, I'll use this notation here where I use S of Z or T of Z to denote the either observed or the counterfactual values of the surrogate and the true outcome under the treatment level Z. And I'll be focusing on when treatment is a binary variable. So the methodology that this work is based on is principal stratification. So as we've heard, there are different methods that have been proposed to do this validation process. As we'll see, principal stratification uses all of these potential outcomes, S and T, as I said, under a binary treatment, and incorporates different modeling and causal assumptions in order to define a quantity under which we can say, yep, the surrogate is valid for future use, or no, the surrogate does not look very promising. We should not use it as the outcome in a future trial. So the principal stratification framework, the idea is that we're going to use these potential outcomes and define an estimand or causal effect. And in particular, the idea is that the causal quantity is going to be conditional on strata. So that's the idea of the principal stratification. And in this case, the strata that we're conditioning on is defined by our potential outcomes of the surrogate. So here, S1 and S0. And a way that we can think about this is that it represents a quantity that is different from just looking at correlation or association. Instead, we're defining causal effects. So in the case here, we're talking about the relationship between the causal effect on S. So I've just given an example. If we just think about some contrast between S1 and S0, that tells us about a causal effect in terms of potential outcomes. The relationship with that and the causal effect on T. So a similar quantity where we have some type of contrast between T1 and T0. And if we can actually understand the relationship between these two causal effects, this would tell us, in essence, whether or not the causal effect on S is more or less predictive or tells us something about the relationship of the causal effect on T. And we're claiming that that's the type of information we need to know in order to say that the surrogate is useful in a future trial. So this framework has been developed under different types of outcome types and in certain settings using covariates. And so here I'm going to be talking about extending the methodology for the case where we have time to event outcomes and we want to include covariates. So we need to make sure that our causal effect quantities are defined that will be proper for these types of data. And after we've defined the quantities, then we'll have to go through an estimation step in order to say for a given data set whether or not the surrogate is valid for future use. So the motivating study here is a cancer clinical trial, specifically prostate cancer. And in this trial in particular, men were randomized to one of two therapies. In both cases, they were given radiation under both treatment arms, but in the second treatment arm, they were also given anti-androgen therapy in addition. And the surrogate that we're interested in evaluating is time to distant metastases. So essentially cancer spreading beyond just the prostate, potentially to other organs in the body, unfortunately. And the overall outcome that we're interested in, or the true clinical outcome, is overall survival. So dealing with time to event endpoints is complex. Of course, we first have censoring, meaning that individuals may or may not actually have the event of interest during the trial. And we have this additional concern when we are dealing with two time to event endpoints known as semi-competing risks. So the idea that it's possible to have both of the events in a certain order. So an individual might have distant metastases and correspondingly have a death event afterwards, but the opposite order is actually not possible by definition. So we want to account for this very particular structure in our data, in our defined models and methodology. So here I'm just starting with some existing approach that was available at the time when I started this study. It's a bit related to mediation, but based on this idea that was talked about with proportion of the treatment effect being explained. And so here I just have a little diagram to help conceptualize the idea that we have our treatment effect can lead to the surrogate, which here is distant metastases, and then our true outcome, which is overall survival. And based on this existing approach, what we found was that the surrogate distant metastases provided 44% proportion mediated of the treatment effect, on the treatment effect of overall survival. So, this is a different method than principal stratification, but we use this as preliminary data to think that this distant metastases is giving us some information about the treatment effect on overall survival. So we were interested in, again, using this as an early indication and then doing further methods development in the principal stratification framework in order to see if we could expand the principal stratification framework for this semi-competing risk time to event setting. So our goal was to define new causal estimands, which I'll call delta S and delta T, based on corresponding models, but not using this proportion of treatment mediation. Still, we had to work with this data setting where we have semi-competing risks. So what we did was we also took an illness-death approach. So here, this is based on the idea that distant metastases and overall survival can be thought of as states in a multi-state model where patients may experience different outcomes at different times throughout the study, and the rates at which they experience those can differ, and even which, if any, of the events they experience might differ from individual to individual. So our models will be based on this structure, and in particular, the data quantities that we're thinking about here are what we can call the gap times, which is the time in between the states. So the illness death model has been well developed in different settings. What we propose to do is to take the illness death model and make it counterfactual, which means we have now a counterfactual illness death model under what we'll say treatment equal to 0, which in this case would be radiation alone. And then we also have a counterfactual illness death model, which is like the potential illness death model we would have under treatment equal to 1, which in this case would be radiation plus anti-androgen therapy. So we basically doubled the number of illness death models that we're interested in estimating. So we want to use our data to try and estimate different parameters in these illness death models. Since we have time to event data, the observed data is going to be a collection of those gap times and additionally our censoring indicators, which I'll call delta T and delta S. So this is basically showing here our collection of observable data that's going to be going into our frailty models. So let me jump into the frailty models. The way that we measure the hazard or the transition intensity is based on these models here. And I just want to highlight this is really the composition of a baseline hazard and then this exponential piece, which is a functional form that allows for us to include frailty or subject-specific terms and patient characteristics through this X transpose beta term. So the idea here is that we want to model the transitions between each of the states in our multi-state models and allow for subject-specific terms and allowing for patient-specific quantities to be estimated. I'll briefly talk about some of the assumptions that we made with the frailty parameters. So a few assumptions of the model here, we assumed that the baseline hazards follow a Weibull distribution. So again, Weibull parameters, scale and shape for each of the transitions between the states from baseline to distant metastases, metastases to overall survival, and from baseline to overall survival. How we decided to do estimation was using a Bayesian approach, using MCMC for its flexibility and incorporating all of these model parameters from our illness death models. We took a pretty general approach as far as prior distributions that I've shown here, assumed vague, non-informative priors whenever possible, which was the case, for most of the model parameters. The part of the model that's much more prior driven that I won't get too in depth here is how we chose to estimate the frailties, because this is really where this counterfactual modeling came in. In this case, we decided to make some assumptions about the frailties and assume that they followed a normal distribution and did set some of the non-identified parameters to set values and use sensitivity analysis to look at the effect of that. But once we set up this whole Bayesian estimation scheme, we're able to estimate the illness death model parameters using Metropolis Hastings. Here I'll just briefly say, estimating the counterfactual frailties did require some modeling assumptions. As I said, we had to fix some of the non-identified correlations in order to get proper draws of our frailty terms within each of the transition modeling. So here I'm showing the surrogacy validation metrics that we had to define for our time to event data. So just briefly, how we decided to redefine what I'm calling delta S and delta T, which is our causal effect on each respectively, is by taking a ratio of the log cumulative hazard. Again, this is a functional form of baseline covariates and our subject-specific frailty terms. And also on the delta T axis or the scale, we decided to use difference in survival probabilities, which was also conditional on all of those patient-specific characteristics that we wanted to include. Let me jump ahead and go ahead and show what these quantities look like once we've done this whole estimation procedure. So how all of this comes together is we develop what's called a causal effectiveness predictiveness plot. So I'm showing those delta S quantities on the x-axis, which is this complex form, these delta T quantities on the y-axis. And the idea is that, as I said, we want to understand the relationship between the causal effects on S, delta S, and the causal effects on T, delta T. So we can look at this kind of plot to understand the relationship between these two quantities. As I said, we wanted to include patient characteristics. So we did some of this plotting where we looked at the effect of delta S and delta T, but now specifically focused on what the effect was based on different patient characteristics. So here I'm showing what this causal effect relationship looks like, conditional on different values of PSA, or prostate-specific antigen. And what we were hoping to find was perhaps different subgroups would appear, where we would be able to identify a stronger causal effect, perhaps, relationship between the causal effects based on different levels of prostate-specific antigen. What we found in the exploration so far was that for this particular example, we were not able to so far identify different patient subgroups for which maybe the surrogate worked particularly well. But we did find that the method, seemed to work well in terms of the methodology was working, and we were able to estimate the quantities, even if we did not find particular patient subgroups in this example. So I'll wrap up and just say that, the idea here is that we wanted to extend the methodology for using principal stratification in the context of incorporating patient characteristics in this illness death setting. We did develop an R-Shiny tool to do some sensitivity analyses. Of course, we'll say we and other groups have evaluated subject-specific subgroups and other settings. And so, I think all of us are continuing to work on these methods, broad picture, trying to help accelerate clinical trials, make them more efficient, and in turn, help more patients in a more timely manner. And then here are some references, and I'll stop there.
Awesome. Thank you all. And I actually think we do have time now for questions for any of our speakers. If you have questions for each other, there's fun overlap between the people and the concepts involved in all the talks. Alternatively, I think we have another like 15 minutes. So Emily, Emily H, if you wanted to show us your real data, we have time for you to do that.
Wait, sorry, could you say that again? Real data?
Like you had a data application that I didn't think you got to show because of time.
Oh, I feel like I said most of the stuff, but I actually maybe I could ask other Emily some stuff.
Yeah.
I actually remember you gave a seminar at our department once. But I was kind of curious, like, could you maybe say a little bit more about why you decided in your modeling like page, sort of the models that you decided on, like, and the parameters that you said you had to um that weren't as that weren't identifiable. Like what ended up happening with that specifically? I'm kind of curious.
Yeah, I'm glad you remember when I visited. It's always nice. Yeah, so, for the context of these time to event models, we kind of had to take a much different approach than some of the other work that I did. Previously had done a lot of work when things were continuous, which of course things work out a lot nicer analytically. When we can make some like multivariate normal assumptions. And so, a lot of the quantities that we decided specifically for the cancer clinical trial were based on the fact that they had to be redefined in terms of hazards is the scale that we went on. Like making some of these assumptions about Weibull distributions. But kind of more to the question about non-identifiability. So, I mean, this is a question that comes up like for any type of outcome, I would say, within principal stratification. And, I would argue it comes up probably for almost every problem in surrogate validation. It just might appear slightly differently. So in this case, the kind of non-identifiability problem, we isolated it to the correlation of our different frailty terms is how we decided to connect the counterfactual quantities. And so in the case here, what we did was we fixed the correlations that represent the joint distribution of the counterfactual things. And we fixed them, and then we just varied them over different things. In other cases, I had put a prior distribution over them, which is one reason why I really like the Bayesian setting. But just due to the complexity for this problem, we decided to just fix them and then vary them to look at the sensitivity.
Great. Any other questions? If not, this was a great session. Thank you both. I'm gonna run to another session I'm supposed to chair, But great seeing you both. And are you all set up for the next session? You have hosting privileges, right? So you can promote people as you need to.
Yes, thank you.
Great. All right, then I'm going to go.
Thanks,
Bye,
Sarah.
everybody.
Bye, Emily.
Thank you. Bye.